Especially that too during COVID time, you all have done your classes on computer, right? Okay. So you are more happy with working on computer or working on a smartphone? Smartphone is better than computer, right? Computer is better? Okay. Why? Good, very nice. You all are wonderful, kids. Everybody have a lot of answer, like because it's not portable. Another guy told me that they have multiple tasks you can do on computer, right? Big screen, right? So lot of facility over there. Okay. So okay, now just this, 1, 2, 3, 6 rows. Tell me, you are interested in biology, mathematics or computer? I divide in 3 groups. Okay, so okay, fine. So if you are in math, what you want to become? Teacher of math. Wow, have a clap karo bhai. Why? Artist. Okay, you? Scientist. Okay. Okay, now no more query. Now, one thing you have to promise with me. Okay? If I'll ask someone to give answer, only they can give my response. Otherwise, no. Okay? Just stay with me for 10 minutes. Okay? And then I will show you whether you like mathematics, whether you like biology, whether you like computer science. We all can come together, okay, whatever interest we have, and we can resolve one of the most required problem of present day scenario, right? Fine. So what we need? We need a complete teamwork, and the most recent and successful example for that was COVID vaccine. You all aware? And many of you who have crossed 12 years age must have taken at least 1 or 2 doses. Nice, perfect. And at least your parents have already done 2 doses, right? So that was the complete and successful example of what? Teamwork. Okay, you must have done many of the experiment in related to the physics, related to chemistry, related to biology, related to some of the activities related to mathematics. Here what I am going to tell you, you will find all subjects together, and that too you are not going to use any chemicals, any sophisticated instruments, or any what we call no chemical, no instruments, but yes, the most powerful computer. And of course, to execute the idea, we need man-made computer, right? And we all are using every day, every moment, right? Okay. So here what I have planned is biology meets computer. Now the question is, how computer is going to help in biology, right? Tell.
And so biology and computer goes hand in hand, I guess. So most of the work done nowadays, the test we take, blood test, anything, that also is to calculate that data, everything, that data collection also happens on it.
Very good. So just I will make this story short. I will start with simple biology. We human being, how many cells we have? Trillions. Okay. So what is the basic unit of life? Okay. So do we able to see the cell by naked eye? What do we need? And I always say one pet sentence to all the students, you know, the things which are visible via naked eye, definitely, otherwise you are not going to enjoy. I am just going very slow, I am not going to tell you anything extraordinary. You all know, but I will just try to make it more interesting. Okay? So, you all know cell, we have already reached to the nucleus. Now, the question is, like you and your sister, you and your brother, or you and your father, what are the similarity? Yes, white t-shirt, tell me.
Sir, we have the genes of our parents and
no, that is later. In visually, what is similar?
Sir, it can be our face.
Face, okay.
Sir, our habits.
Habits. And you also know this is because we are sharing same gene pool, right? So it means all these features are basically controlled by gene. Now, anyone, second row, tell me what is gene? Gene. Yeah. Gray t-shirt, yes.
So it is the DNA, and we have 23 of our there are 23 chromosomes, and each of them contains a special aspect of our, like of its, you can say that if our mother's or our father's eye is blue.
I'll just make it short.
Sir, chromosomes contain genes.
Yeah, okay, young lady first.
Sir, chromosomes contain genes, and genes like help in the transfer of from parents.
I'm very happy, young lady and gentlemen, at your stage at least you know this, right? Yes, you tell me.
So genes are the habits transferred by our parents.
So now I'll just, whatever you said, wonderful answer, but what I'll just trying to, you know, reformat it again. In what manner? You say that gene is basically the part of what you say DNA. I'll just make it functional fragment of DNA is gene. Functional fragments of DNA is gene. Is that clear? Peace. Yeah, I say functional fragment of DNA is called gene that controls each and every trait's characters. Sometimes one gene responsible for one trait, sometimes one gene may responsible for many traits. Sometimes many genes come together and control one trait. Characters, features, fine? Got my point, Peter? Okay. Yeah.
So question. So you said that we get our genes from our parents. So can we get a gene completely ours, not from our parents or our grandparents?
No.
It has to be from ours.
If you want to have a tree, is it possible without seed? That's simple answer. So the genetic constituents we need from parents or from any source. Of course, parents, right? So we need genetic source. Why we need? That's what I'm trying to tell you. When you say you know the nucleus, within the nucleus, you already told me that, sir, there are 23 pairs of chromosomes, right? And you yourself said, sir, on the chromosome there is genes, and then you already understood that each gene are responsible for so much of characters. Now here comes the important aspect. You all know genome? No problem. So when what you say gene, basically gene is what? This is it made up of nucleotides, sequences. What is nucleotide? Nucleotide is basically a base pair. I'll let you know. Like when you read English, how many alphabet you have? When you say Hindi, how many you have? So to read our genetic makeup or arrangement of these genes, we have only 4 alphabets. Imagine. Okay? Yeah, good. Just you know, you are senior school, I think probably 9th or 10th. 9, that's why you know. But I have to take these guys all together on same boat, right? Yeah. So that alphabet is A, T, G, C. Adenine, guanine, cytosine, thymine. Again, for the younger class, I am trying to again take you to the same place where I have stopped talking. You say we all are made up of cells. When you enter cells, the control center of the cells is nucleus. Within the nucleus, there are 23 pair of chromosomes. What we are talking about human. But if you say, sir, what about plants? What about elephant? What about monkey? These number will vary. Okay? So this number is constant for each and every organisms. If there is a variation in this number, things will change. Is that clear? So now you have 23 chromosomes, and on these 23 chromosomes, whatever feature you say, like movement of my hand, color of my palm, hair, my height, my voice, everything is basically indirectly or directly controlled by these genes. Now you must have, I mean, some way or other way, your teacher must have told you about DNA, right? So DNA is basically genetic material, hereditary unit, that's called gene, right? So that is, so when you write your one sentence, many of the alphabets you come together and make a words, and many words in punctuated manner, when you put them together, then it will be meaningful, right? So that's why that gene, I say functional, when it becomes functional, when this alphabet A, T, T, G, C, A, T, so there is a particular orders, right? And those orders is basically code for a particular molecules, regulatory molecules, okay? And ultimately that control everything in our body. I hope this much is fine. Okay. Yeah. Yes, I'll tell you. I'll just hold on. I will show you amazing movie also.
So that is in every cell we have all this.
Yes, every cell, each and every cell.
So in every cell we have the same code that all we have a different code.
No, in one, see, we have two type of cells in our body. One is called somatic cells, another is called reproductive cells. Okay? So in somatic cells, that is responsible for the rest of the body, and reproductive cells is only responsible for reproduction. Yeah. Yeah, I will. To know the sequence of your whole genome. What is genome? Genome is basically the haploid pair. Haploid pair means 23 chromosomes, the genes on the all genes on the 23 pair of chromosomes. If you have the arrangement of these molecules in one together, that's called genome. Got it, right? Okay.
So as you said that there is some sequence to these like adenine, cytosine,
Yeah.
guanine. So sir, if this sequence gets included by another, like instead of thymine there is adenine. So sir, is there a mutation or there is a more likely a chance of getting a disease in that person?
Actually, the problem, very nice question, but the problem is, if you could ask this, because all those questions what you are planning, I have answered. So just wait for, I told you, just keep listening for 20 minutes. And I may be able to answer all your queries, and even if it left one, I will hear for next 3, 4, 5. Till your answer will get your answer, I am here, if sir permit. Okay? Okay, so let's start. What do you mean by wet lab and dry lab? Yeah, orange.
hands-on experiment with chemicals, but then whereas in the dry labs we work on computers or desktops.
Wow, wonderful. I was planning, I should say in vitro, I should say in vivo, I should say in silico. That's the correct scientific terminology. But I thought make it more simple because you may not understood, but you are such a wonderful kids, you already know it. Okay, I am very happy. So yes, he is absolutely correct. Once you say wet lab, basically we are talking about the things which we can do in the test tube, that's called in vitro. When the things we are working on animals or animal system, that's we call in vivo, right? And things which we are doing on computer, that's called in silico, that's the part of dry lab. Why we say in silico? Silicon chip, every computer has silicon chip, right? So that's why we say in silico, right? So now you can see, first one. So yeah, so you can see as you say sir sequencing, I can take plants, I can take virus, I can take bacteria, I can take any animals, right? And then you can collect the sample, extract DNA from that, okay, and then we can see under microscope, right? Yeah, okay, then we can see under microscope, and then even after microscope you have some picture, but you don't understand what is that, how we should analyze. It's like same, yes, so it's the same when you go for MRI, when you go for X-ray, you have a film in your hand, but you don't understand, right? So there you need a great analysis instruments, right? And there comes important role of computer. And sometimes even after computer, you have some this kind of data, but you are unable to understand what is it, right? So for example, we are talking about with the young lady regarding sequencing, right? So when you say sequence, you have the sequence, I will show you sequence, and then I will, what is this? You are unable to read that. But to read that, you have to understand how to read that. And when that's called coding, right? When you have the code, you should know how to decode it. Then the real meaning comes in front of you. Okay, so go to the next. So here comes what is bioinformatics. So bioinformatics is basically when you use these information technology or computer science knowledge and try to decipher or analyze your biological problems, right? That's called bioinformatics. So bioinformatics is not one field. You need computing, you need managing big data, you need collaboration with the community, because first you have to understand what was the problem you are supposed to decipher. Problem solving, you have to think how to resolve it, and then develop a particular diagnostic tool. Right now you say you have COVID, so go for RT-PCR. How it has been designed, right? So first you have to find out so start with this point. First point is to collaborate with the community, communicate. Then you have to find out. Then and then here comes, if I will write A, C, T, D, G, anything, nothing comes. What is it, man? Okay, so everything is there, but we are unable to find the solution. We are unable to take out some take home message. What is it? Nothing. But yes, so what is happening is when you go for sequencing, and if I wanted to know the genome sequence of human being, it is 3 into 10 raised to the power 9 base pair. What is the value mathematically? 3 into 10 to the power 9. And if I ask you to write A, T, T, G, C, all these things, You know how much book, how many size of book you need? You need 10,000 of book. Each book should be of 1000 pages. Both sides you have written. Font size of 12. This much data you have. And this data is what? This data is those 23 chromosomes on which those gene sequences are there. So, we have now all together. You are not getting 1000 and you have 10,000 page.
So that would be for one cell, right?
Yes, only 23, not all, also one cell have 46 chromosomes. I am talking about only haploid set of chromosomes.
What about diploid then?
No, because that is just copy, no. 23 pairs. So, pair is almost copied. So, I am taking one set and out of that this much data generated. Just hold on, just hold on. I just wanted to push you to a particular place and then I will give you chance to ask the question. Just hold on first, go with me first. Okay. So, now you have this much of data and I will tell you, hey gentlemen, you just go and find out a gene which is responsible for your beautiful design of your pinna ear. This is called what you are going to? This is called pinna. Right, this is called pinna. Okay. So, and everyone have different design. Have you observed? Someone have this type, someone have that type, someone, okay. So, if I will ask you to go and find out the gene, for example, hypothetically I am telling you, maybe on the chromosome number 19, I am just giving you. No, I will tell you what is the difference. The things, because we are almost 99.999% similar. That is why we all are human. You are like your parents, you are like your siblings, but I have given you a value. You all know good mathematics. What is that value? 3 into 10 raised to the power 9. So, if you calculate a value that is 99.999, it means the difference of sorry difference of 0.001, still you will find almost difference of approximately 1 lakhs base pair. Once I say base, I am talking about alphabet. Now you imagine you have a book or you have a library, rather I could not say book, 10,000 book is not book, it is a library, and in this library this much difference. So, why you look different, I am looking different, is reason being the difference in these alphabets which I have told you the values. Now, that young lady asked, sir, what will happen? We have, this is the difference, but the question is if there is a one sequence change, one alphabet change. Answer is sometimes one alphabet change makes no difference, but sometimes that difference makes lot of changes. Here I will show you how. First I will just try to explain you that those sequence imagine here, and then you take all those features. Do not worry about what in what way I have represented here, but the story which I have told you, that is most important. Okay. So, these are the representation. This is the ask her to bring that bottle. So, the raw data, this raw data is now you have in sequence form, they have different features because different chromosomes have different information. Now, you pass on to, and you know that whole complete stretch is not the functional one. In between the break, there are some of the functional and some are non-functional. So, the functional one we say exon, non-functional one is called intron, but that is also valuable. It's not that if it's not functional, initially people say junk DNA, but now nothing is junk in our body, you know, nothing is junk. If anything is junk, that start creating problems. So if it is not creating problems, it means it is very, very important, and they play a role in regulatory one. Okay, so now back to the basic. Okay, you want to see some movie? But the question is, you should just focus on that and keep looking at that, and in between if I will say something, you just follow it. If you disturb in between, then I will show you movie at the end. Yeah. Yeah, you keep answering, don't waste time. Yes, every cell have 46 chromosomes. In our case, 23 pairs I say. Yeah, except one, that's called sex chromosome.
So that is all in one cell.
Yes, one cell. So imagine we have in each cells.
So in each cell we have all of those.
Yes, and this much data we have each cell, and imagine all those are working in coordination, then only we are talking to each other. I'm not getting your question.
So you say that every
Eye cell, each and every cells. So your eye is made up of one cell? It's millions of cells. So but each cells have because you know cells coordinate become tissue. Tissue coordinate become organs. Organs coordinate become organ system. Organ system coordinate become body. So now try to understand. I got your question. So because each cells are coordinating with each other and then controlling one. No, not the chromosome. Not the chromosome, gene.
every cell of our body. In every cell. So even that the cell that is in my
Sir, it is like in your computer, you have 50 components. If one component will not work, is it able to computer will work? So each cell is a system like computer, more than a supercomputer, and those all supercomputer work together and come out with one output. That is your eye. Got my question? Okay. Now just hold on. The most important part, if you understood this, my story will be very easy to tell you. This is called central dogma of molecular biology, or what we call central dogma of biology. It means, I say, you imagine that your cells is like a building. Within this building, this is Vigyan Bharati office, but Vigyan Bharati chief, for example, Dr. Kapil sir is chief. So that chief is who? Chief is this nucleus, DNA. Now if you want water, sir is not going to ask, sir is not going to give you the water bottle. What sir is going to do? Sir is going to ask maybe the canteen owner, and this person will ask some of the helper to provide you water. So ultimately you want water, who gave you water? Third person, workers, and ultimately you get it. So here imagine in our body the chief manager, head, DNA gives some, you want water, but he will say in other way, in his own language. The language is chemical language here. So they pass this information, DNA to RNA, and then RNA to protein, and ultimately protein is the main regulatory molecules that you all know maybe in the form of enzymes. You all know our membranes is made up of lipid bilayers and proteins. Okay, there are many hormones, many enzymes, fibrous proteins, everything. Building blocks of your body is what? Protein, right? So ultimately this is executed by proteins. Now I am just telling you, sir थोड़ा सा डिले करना पड़ेगा हमारे सेशन को. हाँ, एक बार आप ये दिखाओ इनको. अब इसको ध्यान से सुनना, आवाज नहीं करना है. और इमेजिन करो कि नाउ यू आर एंटरिंग टू द सेल न्यूक्लियस. यस, दिस इनक्यूजिटिव बॉय, आप ध्यान से सुनना इसको. ओके, गुड. और इसके बाद फिर मेरी स्टडी आसान हो जाए. ये अगर समझ गए आप, मेरा बहुत आसान होगा. आवाज है? No problem, I will with you. So now we are entering nucleus, inside the nucleus, okay, and these are the chromosomes are located inside the nucleus, and on the nucleus you will find this genomic DNA. These are genomic DNA, okay. So this DNA is basically when the chromosomes initially they are like this, so they start unfolding, and then you will find these are the DNA structure. This is a model, DNA model, and this contains lot of information, but this information is basically transcribed into RNA, okay, and then RNA will pass on this information, and that is called translation into in the form of called protein that I have told you. Now this is the first step, that preparation of how they will transcribe into RNA, that you try to understand. What they do? They gather on the DNA, these are DNA. So one of these, this is called the factors. You need lot of protein molecules, which I say regulatory molecules again, which is inside our body, inside the nucleus. They will go and they will catch on this DNA, this stage is DNA, and this is the machinery which is going to attach with that. Look, this is also again application of computer. If you know the computer, you are able to make this. Okay, so now this everything is happen where? Inside your nucleus. So each transcription factor has different roles. These, when I say factors, mind it, it's a protein which is already produced inside our nucleus. Okay. Now, not inside our nucleus, produced in our body, and it will enter inside the nucleus and play this responsible role. So this long, and then they keep adding, keep adding, keep adding. That's called C-terminal domain, CTD. So now RNA transcribe from DNA, and then this machinery will leave, okay. Yes, now they are broken. So using the aggregate transcription factor as scaffold. These are the enzymes. Again, all enzymes are, most of the enzymes are proteins. Like Hollywood movie, right? So you are making this again application of computer in biology. This work itself, got it? So the role of this transcription factor is undo the double helical structure of DNA into single helical structure. So break those bonds and complete their work. So one part is over, that transcription of the genomic information. Now transcription means you have, instead of DNA, you have a complementary copy of RNA. I'll just stop, just finish it, then I will come to this. Otherwise, in between you are getting diversion from my voice and that, right? Okay. These all materials are already available in our body. When you say you are eating nutrients, you are eating vitamins, you are eating all those things, that makes all these materials which is required, and that's why. And when you make 1 cells become 2 cells, 2 cells become 4, 4 become 8, 8 become, so everything happens because this is happening. And now you have, they have the bend, and now this is the stop. Now you have already RNA. So, because in bacteria they don't, all the complete sequence of bacteria in prokaryotes we have exon, they don't have exon intron. In case of eukaryotes we have exon and intron. So, exon are the functional part, which I already told you before. I think they are more, after this we can start the activity, so that they could understand. Or if you want, I can share this link and you can watch at home, right? Even during night you can see at your home, that's more, right? Confusing, no? Okay. Right. More than one time? Good. Yes. Sure. Oh, no problem. No problem, beta. We can revise and we can again. This you remember, I am using this information. 3 nucleotide base will code codon, that's called codon. That is going to code for 1 amino acids. In our body we have 20 amino acids. I say there is full alphabet A, T, G, C. Combination of these 3 will code for 1 amino acid. Okay? Is that? So, this is the information you remember after this for activity. Can you stop? I am stopping it, but I am sharing with you, and watch it again and again and again, and you will understand. Now, come to the next. Yes, next move. Yeah. So, okay. Yes, this one. So, now just hold on. I have already told you that this is your cell. Within cell, we are watching movie of all the story which is going on here inside. And I say this we have 46, and out of this I have taken out 1, and then if you unfold it further, you will have this kind of DNA and fragment. So this is only 1. So imagine 23. Then I say that the total base pair, if you write A, T, G, C in that format, this is the value, and the difference between each of us is for 0.0001%, and that's the reason of difference of our appearance or traits. Okay, the only difference is, so these all are in pairs. So that's why I say, one of you people say, sir, why we are taking 23? So why we are taking 23? Because these all are in pairs. So 1, 1, 1 from each. But here difference is, if it is a male, you will have XY. If it is female, we have XX. So 23 pair is somatic chromosome, and this is called sex chromosomes. Okay. So now where is the gene? So all these chromosomes, these are the genes. That's called location of the gene on the chromosome, and that is important to know. Now I can move to the next. So this, if you go for the detail, I say 4 alphabet is important: A, T, G, C, adenine, guanine, thymine and cytosine. But when you go for DNA to RNA, there thymine, instead of thymine you will have uracil. Okay. So that is the only difference, but I say all this information will be passed on to RNA, but instead of like A is, leave it. A is going to bind with T with double bond that is hydrogen bond, and if we C is going to bind with G with triple bond. Okay. But instead of T, in case of RNA you will have U. Okay, this is the only difference. But after RNA, when you go for the proteins, I told you that proteins provide many essential functions. Why we need proteins? Again I am coming to DNA, RNA, again coming to protein again and again. The reason being, these all functions you can read here is basically provided by the proteins. And if you go a little bit more structure of the proteins, basically the proteins have 4 type of structure, but to become the functional one we need a quaternary structure. Okay. So, for in general when they produce, that story you are seeing now movie, they will produce a string of amino acids, that is called polypeptide, right. What you call amino acid, multiple amino acids come together will form peptide, huge chain polypeptide and when this become, when the 2 peptide chain will come together, they form secondary structure when they are going to have bind with through hydrogen binding. So, primary become secondary, when further one more bond is going to add with this, start folding, become tertiary, and when 2 tertiary molecules come together they will form tertiary quaternary structure. Okay. And this quaternary structure is very, very important for all function. Till the tertiary structure, protein is not functional. The moment they achieve quaternary structure, they become functional. Now, here now I am going to start. Now you have to play with me, okay, because you do not have laptop in your hand. So, what we will do it here and you just follow and enjoy. And I am not going to ask, do anything difficult one, very simple one. You all know diabetes, you all know cancer, very common disease. Okay. Now, just because I have written 1, so the moment I say 1, you guys you ask, sir, why it is 1, maybe type another type. So, that is why I just give you the glimpse what do you mean by type 1 and what do you mean by type 2. Generally, when you talk in simple, so usually you will have type 2. Okay. So, type 1 and type 2, usually in our society most of the people are suffering from type 2, but usually people take even sometimes juvenile diabetes at younger age, and then you take insulin tablets or insulin injections regularly, right. So, do not confuse. So, just remember there is a diabetes, that is a problem. Okay. So, I am try to decipher where this computer you can use and how it is helpful to resolve these problems. Okay. So, what is happening now? The question is, if I say these are the characteristics, generally it does not mean if someone have any of these become diabetic, no, it is just a symptomatic things. It is going to correlate with the test and then only you can say this particular person have diabetes. Now, what is happening? Why diabetes happen? You all know lock and key. If it is a correct key, lock will open. Incorrect key, it is not going to open, right. So, again insulin is also a type of proteins, it is a hormone, right. So, what is happening? Imagine this is your body and there are receptors. So, if insulin is basically, how does insulin work? If insulin is going to bind correctly here, you need energy, no? To work you need energy, and what is the instant source of energy? Glucose. So, if this insulin is not going to bind with this receptor, glucose is not available for your cell, and there is no glucose, no energy, unable to work, you feel lethargic, you feel laziness, you feel sick, right. So, it is important that insulin should bind with the receptors. If it is bind with receptors, this channel open. When it is open, glucose is coming in. When glucose is coming in, you respire, glucose will break down give the ATP, we have sufficient energy and keep working. So this mechanism is going on every microsecond, mini nanoseconds. It is going on in our body. So imagine, that's why if someone have this problem, so insulin is not performing this duty, or maybe insulin is not available in that particular person, that's the reason of disease. So now it means we need insulin. So insulin and insulin structure is like this. Usually we have, but that is in proinsulin. To make the active insulin, we need to cut this C chain. Then, and this gly, valine, glutamine, glycine, cysteine, these all are amino acids. These all are amino acids. I so told you how protein makes. Number of amino acids make peptides. The number of them, when they try to achieve the quaternary structure, then they become active, and then they start functioning. So type 1 diabetes is caused by basically rare genetic disorder, and we will now use the bioinformatics to better understand the cause of this rare genetic disorder. Yes, next. So now एक्टिविटी 1, अब आप लोग ध्यान से सुनना और इसको देखना कि कैसे कैसे हम करते हैं एप्लीकेशन. अब मैं भी बोला था ना आपको कि यार ये ATGC ऐसे करके लिखे, ये मीनिंगलेस है ना ये सेंटेंस. देखो पढ़ के कुछ मीनिंग है क्या? कोई पढ़ सकते हैं इसको? यस. but there is no meaning, no? That's what I'm asking. Yes, but how? If you take these three together, if you take 3 code, and I will tell you which 3-letter code is going to code for which particular amino acids. So imagine 3 this base will code 1 amino acids. So every 3 1 amino acid, every 3 1 amino acid, every 3 1 amino acid. You have multiple amino acids. Now you have peptide. Multiple amino acids come together form peptide, and then when they achieve their folds, they make bond in between, they are going to form protein, and that become the active one. Okay, now you start. Yes. ATGC, adenine, guanine, thymine, cytosine. अभी आप सो रहे थे मुझे लग रहा है. कोई बात नहीं. ओके, अब आप देखो मैं क्या कर रहा हूं. अब ध्यान से अभी आप देखना है, ये मैं आपको शेयर करूंगा और घर में जाके करने का है. ठीक है, मैं अपना ईमेल दूंगा, फिर मुझे बताना किस किसने किया है. फाइन. अभी देखो हमने क्या किया, वापस चलो एक बार फिर से. ये आपको टास्क है, ये मैं आपको मेल पे शेयर, अभी सर आपको सबको भेजेंगे व्हाट्सएप पे. आपने क्या करना है बच्चे, ये जो ये है इंसुलिन को, इंसुलिन में क्या बहुत सारे पेप्टाइड होते हैं. तो यहां से जो C से लेके C तक है, इसको हमने एक फ्रैगमेंट लिया एंड आई एम गोइंग टू कॉपी इट. कॉपी करके जैसे आप लोग गूगल सर्च में जाके इंजन में डालते हो ना, ऐसे एक हमारे पास टूल है, जैसे वीडियो गेम करते हो आप, तो ब्लैट, उसका नाम क्या है, ब्लैट. आप जैसे इसको क्लिक करोगे या आप गूगल में लिखोगे ब्लैट और क्लिक करोगे तो एक ओपन विंडो ओपन होगा. दिखाओ भैया विंडो, ये विंडो ओपन हो गया. अब जो वहां कॉपी किया था ना हमने मीनिंगलेस सा सेंटेंस इंसुलिन के लिए, उसको लिया और यहां पे हमने पेस्ट कर दिया और पेस्ट करके सबमिट कर दिया. अब देखो क्या करेगा ये. अब जैसे सबमिट किया तो इसने एक अजीब सा रिजल्ट दिया आपको. अभी तो हमने क्या दिया था, एक मीनिंगलेस सा ACTGC करके डाल दिया था. अब उसने क्या बता दिया मुझे, आपके जो सीक्वेंस थे कितने अल्फाबेट थे उसमें, 60. स्टार्ट कहां से हुआ था, एंड कहां पे हुआ, साइज कितना हो गया. अब ये कहता है आइडेंटिटी 100%. ये क्या मतलब है, ये कह रहा है कि इंसुलिन तो ऑलरेडी नोन है ना. मैं तो आपको क्या बोला, मैं एक सिंपल एग्जांपल दे रहा हूं. अब जैसे ऐसे कोई और नई चीज़ों के बारे में अगर हम करेंगे तो हो सकता है ये 100% की जगह हमें 90% देता, 60% देता, लेकिन ये तो नोन है इंसुलिन. इसीलिए जैसे इस मशीन के अंदर हमने ये इंफॉर्मेशन दिया, उसको 100% दिखाया कि इसकी आइडेंटिटी मतलब सिमिलर, बिल्कुल मैच कर रहा है. तो आपको यहाँ पे 100%. अब सवाल ये है कहाँ पे ह्यूमन बॉडी के अंदर, ये जो हमारा सीक्वेंस बताया ना इतने सारे बेस पेयर, वो ऑलरेडी सॉफ्टवेयर को पता है। 3 × 10⁹ वो ऑलरेडी है। अब ये जो छोटा सा फ्रैगमेंट मैंने लिया, अगर मैंने बोलता कि जाओ और लाइब्रेरी से वो बुक में से ढूंढ के लाओ, कहाँ मिलेगा? तो पूरा 1 साल लग जाता ढूंढने में ही। कहाँ से ढूंढते? सोचो 10,000 बुक, हजार पेज की बुक है ना, पढ़ो बैठ के साल भर, कुछ नहीं मिलना। अब कंप्यूटर ने क्या किया, अभी आपके सामने दिया और हमें क्या बताया मालूम है? कहाँ पे है? सी क्रोमोसोम 11, जो 23 क्रोमोसोम थे उसके 11 नंबर क्रोमोसोम पे ये इंसुलिन वाला जो जीन है वो प्रेजेंट है। अब बताओ कितना आसान हो गया कंप्यूटर से? नहीं हो गया आसान? बताओ 10,000 बुक ढूंढते और कितने सेकंड लगे? मुश्किल से 2 सेकंड भी नहीं लगे, है ना? तो इसीलिए तो करना है। अब ये तो सोचो अब तक है, आप लोग सोचो इससे फास्ट कुछ करके दिखाना है। यही तो काम करना है कंप्यूटर से। तो बायोलॉजी तो है, लेकिन काम किससे कर रहे हैं हम? कंप्यूटर से। कितना आसान कर दिया ना काम को। अच्छा, अब क्या है इसका जो पोजीशन है? अब सवाल है कि सर आप जो कह रहे हो कि वो जाकर के 11 क्रोमोसोम है, लेकिन 11 एक क्रोमोसोम में इतने सारे सीक्वेंस थे, तो उसकी पोजीशन क्या है? देखो वो भी नंबर आपको बता रहे हैं, इस पोजीशन से लेके इस पोजीशन तक। तो इतना स्पेसिफिक इंफॉर्मेशन 1 मिनट के 1 सेकंड के अंदर आपके पास आ गया। नेक्स्ट, स्पैन 60 मतलब 1 पोजीशन से लेके 60 पोजीशन तक था, उसको स्पैन बोलते हैं। इस पोजीशन से इस पोजीशन तक स्पैन, ठीक है? ओके, अब दूसरा एक टास्क और लो, कोई और। तो आपने क्या देखा इसमें क्वेश्चंस? ऑन विच क्रोमोसोम 23 क्रोमोसोम वाज इट वाज ऑन, एंड व्हाट वाज द पोजीशन ऑफ दैट जीन? आपको ये नंबर बता दिया, दिखाया था ना उसमें। तो ऐसे ही क्वेश्चन मैं आपको भेजूंगा, आप मुझे आंसर दोगे, ढूंढ के करोगे ना? ओके, नेक्स्ट। अच्छा देखो, अब ये कहते हैं कि सर डायबिटीज सबको तो नहीं होता है, ऐसा तो होना भी नहीं चाहिए। अब होता है तो डॉक्टर कैसे पता करे कि ये इसको हुआ तो अगले जनरेशन में नहीं होगा, या सच में डायबिटीज है? कैसे इसको डायबिटीज हो गया? क्या इसके पापा को था, क्या इसके दादाजी को था, या उसके दादी जी को था? कैसे पता चलेगा? तो उसके लिए डॉक्टर क्या करता है, एक एनालिसिस कहता है, उसको कहता है पेडिग्री एनालिसिस। अब ये